today to introduce today's speaker, Patrick Burke. Patrick is a native of Gaithersburg, Maryland, and he did his undergraduate here at the University of Maryland, although he first got a degree in geography and then came back, saw the light, came back and got his degree, a second bachelor's degree in environmental science and technology. And while he was an undergrad in ENST, he was also a member of the soil judging team. And then the last couple of years, he's been serving with me as assistant coach for the group. And so he was a very helpful, important part of the Terps victory 2 weeks ago. And then just a couple of other things. Patrick is also an avid birder. You know, I'm always asking him, you know, what was that? You know, he always has the answer. I don't know if he really knows, but he sounds like he does when he tells me. And he's also quite the videographer. And so when he was doing some field work at one point, he said, well, you know, I could make it, so, you know, it'd be interesting to see how those coring and all this stuff is done. He hasn't had time to kind of fit it into today's seminar, but he could have. And they did this really nice video of them collecting cores out in one of these wetlands. So anyway, so he's a multi-talented guy. But today he's going to be talking about part of what he spent the last couple of years on, working on his master's work on carbon stocks in depressional wetlands on the Eastern Shore. So, Patrick?
So there actually will be some stills from that video in the presentation. So thank you for being here today. I'm very excited to talk to you about one of the projects I've been working on for my master's work, which is looking into optimal sampling strategies for obtaining or estimating soil carbon stocks in Delmarva bays. And believe it or not, each one of these circular features on this DEM map of a region of the Delmarva Peninsula is a Delmarva Bay. More on Delmarva bays in a little bit. So I'm sure everyone here has seen a diagram of the carbon cycle that looks something like this. I'm not going to go into an incredible amount of detail about this. This is just here to show that this project is about a very small but important part of the carbon cycle, which deals with the interactions of soil and the atmosphere. Specifically, this project deals with how soil can sequester carbon and mitigate against climate change. The main cause of climate change is the greenhouse effect. And the three major gases that contribute to the greenhouse effect are carbon dioxide, methane, and nitrous oxide. Following the Industrial Revolution, a large and growing amount of CO2 exists in the atmosphere. Sequestration of carbon is an important climate change mitigation mechanism, and soils have a lot of potential to sequester carbon, as they're a major sink for carbon. You can see in this diagram to the right, 3 of the main active pools of carbon on the Earth. Soils contain the most gigatons of carbon at around 1550, which is more than both the atmosphere and vegetation combined. Wetland soils in particular contain around 20 to 30% of all of this carbon that's stored in soils and occupy only a small amount of the land surface, about 4 to 6%. So why exactly do wetland soils contain so much carbon? To answer that question, we need to take a few steps back and first define what a wetland actually is. So a wetland needs to have hydrophytic vegetation, which is vegetation that is uniquely adapted to the saturated soils of wetlands. Typha or cattail is an example of hydrophytic vegetation. A wetland needs to have wetland hydrology. Surface water is an example of wetland hydrology. And lastly, a wetland needs to have hydric soils. And this image right here of a core obtained from one of our sites is kind of a classic example of what a hydric soil looks like. Hydric soils commonly have thick, dark surface horizons and gray subsoils beneath. The definition of a hydric soil offers even more insight as to why wetlands store so much carbon. So the definition of a hydric soil is a soil that forms under the conditions of saturation, flooding, or ponding long enough during the growing season to develop anaerobic conditions in the upper part. And it's the anaerobic conditions that are brought upon by saturation is why, or one of the main reasons why wetlands store so much carbon. So what exactly causes a soil to be anaerobic? It's a saturation, but specifically, a soil becomes anaerobic because oxygen diffuses 10,000 times slower through water than it does through air. So when the soil is saturated for a long enough period of time, oxygen is depleted, and then the soil becomes anaerobic. When the soil becomes anaerobic, anaerobic microbes use alternate electron acceptors other than oxygen for their respiration, and use of these alternate electron acceptors yields less energy than oxygen. And this causes or results in the primary electron donor in respiration, which is mainly carbon and soil organic matter, to accumulate because it's essentially used at a slower rate. And this is kind of the main kind of mechanism as to why wetlands store so much carbon. So soil carbon stocks are basically a way to quantify the amount of carbon that's stored in the soil. Specifically, they are just the mass of carbon stored per unit area. They are commonly expressed in kilograms of carbon per square meter or megagrams of carbon per hectare. The measured soil carbon stock value within a wetland can offer insight into a wetland's history, its land use and formation, and its potential for carbon sequestration. If the carbon sequestration ability of a wetland is known with high confidence, then there will be both more incentive to protect that wetland and more accurate accounting of the total carbon sequestration of wetlands. To calculate the soil carbon stock of an observation at a site, you need 3 different data points, and those are depth of each horizon or thickness of the horizon, which is shown here, bulk density of each horizon, and the percent organic carbon of each horizon. So this is pulled from our data set. It's one observation that we obtained. And to calculate the carbon stock of each one of these observations, we first obtain the carbon density of each horizon, which is calculated by combining the bulk density and the percent organic carbon. Then to get the amount of carbon in kilograms per horizon, that is when you factor in the depth of each horizon or the thickness of each horizon. To get the total carbon stock of a particular observation, you simply sum up all of the amount of carbon per horizon. And I should mention that carbon stocks are typically expressed to 1 meter. So soils are inherently heterogeneous, and because of this, soil properties are variable. Here are some examples of different soil properties and how variable they are based on their CVs, or coefficient of variation, which is the standard deviation divided by the mean. Soil color, soil pH, and thickness of the A horizon are some examples of the least variable soil properties. Particle size distribution or soil texture, cation exchange capacity, and soil structure are moderately variable. And solum thickness, or thickness of the A and the B horizon, percent clay, and organic matter content are considered some of the most variable soil properties. Carbon makes up a large portion of organic matter, so these 2 properties are correlated. So generally, there are 2 different kinds of variability in soils. Systematic variability is variability that's gradual and predictable based on changes in landforms. So take a hilly mountain and a flat valley, geomorphic elements going from the top of the mountain or the summit to the bottom of the mountain or the toe slope, and the 5 soil forming factors. This diagram here is a good example of systematic variability of organic matter content. we know that organic matter accumulates more towards the toe slope of a topographic gradient from erosion, from surface material upslope and then deposition downslope, and also from saturation at the toe slope of a topographic gradient. The other kind of variability is random variability, and this kind of variability is highly complex and cannot be related to a known cause. Some say that random variability could be caused by changes that occur on very, very small scales, such as changes in lithology, biological activity, and hydrology. And this kind of variability is mainly what this project deals with, as our sites don't exhibit many changes in landform, geomorphic element, or soil forming factors. So a simple answer to addressing this high variability and to more accurately estimate the population mean of a site is to simply increase sample size. However, obtaining a single observation for the soil carbon stock value of a wetland is both time-consuming and expensive. We estimate that excluding travel time and expenses, it takes around 5 hours of effort and around $200 to $500 in analytical costs to obtain the carbon stock value for a wetland using just one observation or one sample. Because of this, some individuals may report soil carbon stocks from a single observation at a wetland and call it that. However, nothing can be inferred statistically about the soil carbon stock value of a wetland using just a single observation. And even if you double the amount of time and effort, a statistical statement can be made, but with a wide margin of error likely. So all of this kind of begs the question, what sample size is required to achieve a balance between sampling and analytical effort and cost and accuracy of the soil carbon stock value of a wetland? This question is especially important in today's world due to the emerging carbon credit market. So carbon credits are tradable certificates that represent the reduction, removal, or avoidance of 1 metric ton of CO2 from the atmosphere. So they basically add a monetary value to a piece of land based on how it contributes to climate change mitigation, essentially. Carbon credits are used by companies, governments, and individuals to either comply with regulatory limits set by agreements such as the Kyoto Protocol, or just voluntarily offset their own emissions in pursuit of climate goals. So now the carbon storage potential of land has a monetary value, so that makes the accurate quantification of carbon even more important. One of the objectives of this research is to simply quantify the amount of carbon stored in 3 Delmarva Bay wetlands that occur in close proximity to each other along a restoration trajectory. So there's a prior converted cropland, or a PCC, that was drained and used for agriculture and will be restored in the near future. There's a natural wetland that's remained undisturbed, and a restored wetland that was also drained, used for agriculture, and then restored in 2004. So this part of the project is contributing just a small amount to a larger project that is being done by the USDA NRCS that's evaluating the effects of wetland restoration using a before-after control impact study. So the USDA NRCS have installed eddy covariance flux towers at these sites, which are tracking fluxes in greenhouse gases. They're also looking at the hydrology, and then they'll be using our carbon data to inform this study. Another objective of this research is to assess the variability of soil carbon stocks within each wetland type and develop an optimal sampling strategy to obtain reliable soil carbon stock data within a specified degree of confidence for minimal effort and cost. So Delmarva Bays are non-tidal freshwater depressional wetlands that are found throughout the Delmarva Peninsula. However, they're concentrated in the northern part of the Delmarva Peninsula. Prior to colonial settlement, there are estimated to have been around 17,000 Delmarva bays, but post-colonial settlement, many of these Delmarva bays have been drained for agriculture. Delmarva bays have a mean area of 2.83 hectares. Many of them are elliptic in shape and oriented in a northwest-southeast direction. Many of them also contain coarse-textured, sandy outer rims. There has been a little bit of research looking into how these Delmarva bays formed. As thinking back to that DEM image at the beginning, you wonder what had to happen to cause the landscape to be like that. And there have been some pretty wild theories proposed, ranging from asteroidal impacts to large marine organisms kind of creating little depressions on the ground when this area was submerged. But the most likely hypothesis for their formation is that they formed from intense wind blowouts that occurred during the late Pleistocene, when this area was more of a tundra and taiga-like climate. And the fact that these Delmarva bays, or many of them, are elliptic in shape and have this distinct orientation and sandy outer rims are pieces of evidence that support this hypothesis. So our sites are located in Caroline County, Maryland, right by the Delaware border. The prior converted cropland is up here. About 2 kilometers south of that is the natural wetland, and then about a half a kilometer south of that is the restored wetland. Just by looking at this satellite imagery, you can see evidence of other Delmarva bays on the landscape, which is pretty cool. And then when you overlay a DEM map, you can see even more evidence of Delmarva bays. Again, many of these are drained, and you can see linear drainage ditches that are running through many of these Delmarva bays. So the first thing we had to do before sampling was to determine the boundaries of these sites. And that was pretty easy at the natural and restored wetlands, as these sites clearly went from hydrophytic vegetation to vegetation that's commonly found in the upland. But at the PCC site, this was a little more difficult, as this site no longer has its natural vegetation or hydrology. So initially, we determined the boundary by looking at satellite imagery and basically drawing it where the soil surface was the darkest. Then once we sampled close to the boundary, we went out to the field and used something called field indicators along transects that cross the boundary. Field indicators are basically criteria that can be used that are based off of soil morphology that can tell you whether or not a soil is hydric or not. We sampled these sites at a high frequency. We obtained 30 observations at both the natural and restored wetlands. Initially, we obtained 30 samples at the PCC. Then the restoration project was changed, and more of the site was going to be affected by the restoration. So we returned to this site and obtained 10 more samples at this part of the site that is going to be affected by the restoration. So in all 100 of these observations are down to 100 centimeters. Here are all 3 of our sites. PCC is on the left, naturals in the center, and restored is on the right. You can see that the observations are equally distributed throughout the sites. Looking at the PCC, the 30 initial observations were all along this eastern crescent of the site. And then we returned to the site and went on the other side of this drainage ditch and sampled it 10 more times. So we used 2 different methods to sample the soil for this project. For the upper 50 centimeters, we used this specific aluminum cylindrical core. And that's because we needed to calculate bulk density of the soil for the upper 50 centimeters. As I said earlier, one of the requirements for carbon stocks is bulk density. And to calculate bulk density, you need the oven dry mass of the soil and the volume of the soil. We know the volume of this core, so we were able to calculate the volume of the soil in the core. To get the core in the ground, is much easier said than done. Basically, one person puts a board on top of it and another person hammers it with a mallet. One observation we made is that the soil inside of the core would often be compacted after hammering it into the ground, which is expected. So when this would happen, we would measure the amount of compaction, write it down, and then back in the lab on the computer in Excel, we would basically stretch the soil length back to its original 50 centimeter length. And we would do that for the horizons that are most susceptible to compaction, which are the O horizons and the A horizons. So once the core was in the ground, we would dig a hole adjacent to it with an auger. Then we would either take our arms or this fancy metal core remover and place it in the auger hole, then leverage underneath the core to prevent soil from slipping out the bottom, and then pull upwards. So you can see here, the metal core remover is going in the hole, then it's going down, being leveraged underneath the core, and then pulling up. Then we would go down the same hole that the core was in with a bucket auger and sample the soil from the 50 to 100 centimeter depths. For the soil in these depths, we would not calculate bulk density as we did not know the volume of soil that we were sampling. So instead, we opted to use something called pedotransfer functions, which essentially require you to research soils that are found in a similar region to your soil or contain similar soil properties, and then apply those measured bulk densities to your soil. This is a appropriate way to go about obtaining bulk density for this soil in this depth, as the carbon content of this soil is really low, usually at about 0.1%. So it doesn't contribute much to the overall carbon stock value of the observation. So we would sample this depth with an auger, get the soil out of the auger, put it in a gutter where we would describe the soil morphologically and separate it into horizons. And then we would take soil back from each horizon back to the lab. At the lab, we would open and describe the core the same way we describe the soil in the field. We would open the core with a pair of metal shears and then describe the core or the soil in the core based on morphology. And then all of the soil would be oven dried and ground to a specific size to be analyzed for organic carbon content using LECO elemental analysis. So this figure shows the relationship between bulk density and percent organic carbon. And as expected, this relationship is negative. As percent organic carbon increases, bulk density decreases. So I've separated all 100 of these points into different categories based on different kinds of soil that can be found in wetlands. So all of the points to the right of this line represent organic soil, which is soil that contains partially decomposed vegetation and has a high percent organic carbon, greater than 12%. And you can see that all of these points are falling in a pretty low bulk density. Mucky mineral soil is also a kind of soil that can be found in wetlands. This soil is a combination of mineral and partially decomposed vegetation and has a percent organic carbon of 5 to 12. This soil also has a low bulk density, but higher than pure organic soil. And then most of the soil falls in the category of mineral soil, which has a percent organic carbon less than 5 and is dominated by minerals, but also frequently contains organics. Now we're going to look at the relationship between soil color and percent organic carbon. So this image right here that I'm going to point you to is a page from the Munsell Color book, which is a way in which soil scientists color soil. The only thing that you really didn't know about this page is that value is on the y-axis of the page, and this represents how light soil color is. And the lower the value number, the darker the soil. The lighter the soil is, the higher the value. And you can see that soil with more percent organic carbon typically has less value. So looking at the soil with a value 2, which might look something like this, the percent organic carbon ranges from about 1 to all the way up to 60%, which is about as high of a percent organic carbon that you can get. Then looking at soil with higher value, this soil has much lower percent organic carbon from 0.1 to 1. Now we're going to look at the relationship between edge distance and carbon stock at the PCC. Going from about 0 to 30 meters, it seems like there's a positive relationship between the two. So as you go further away from the edge at the site, carbon stock increases. This relationship would be considered systematic variability, as we generally know that the edges of wetlands are going to be better drained, less saturated, and contain less carbon compared to the center, or at least further away from the edge. Beyond 30 meters, however, these points would probably fall under the category of random variability, as we're not quite sure what's driving the differences in these points. Here are all three of the sites. It seems like there's also random variability, but throughout the entire distance from the edge at both the natural and restored wetlands. Now we're going to briefly look at the total measured carbon stock values of these three sites. But I just want to preface this by saying that comparing the carbon stock values between sites was not the objective of this research. But it's worth to at least see how much carbon each one of these sites contains. So the PCC contained the lowest carbon stock at about 17 kilograms of carbon per square meter. The natural wetland contained about 22, and the restored had about 32. It's expected that the carbon stock of the PCC is the lowest, as this one has been cultivated for a long period of time and drained. So the carbon at this site has been oxidized and lost continually. It's not as expected that the restored wetland, however, has a higher carbon stock value than the natural wetland, as this site was also drained and used for agriculture. One possible explanation for this difference is that this site just had a really, really high kind of base level carbon stock value before cultivation. Maybe it was saturated for a really long period of time, with a lot of water, or maybe there was something about the biological inputs from the vegetation that just caused this site to be abnormally high in carbon. So that's one possibility. Now we're going to look at some figures that further explore the variability of carbon stocks at these sites. So this figure shows number of samples on the x-axis and carbon stock on the y-axis. The green line that goes through the center is the true mean carbon stock value of the PCC, which again is at 17. The orange and the blue lines represent the mean max and mean min carbon stock value per number of samples. These values were created by running 20 different scenarios that pulled a different or random arrangement of carbon stock values for each number of points sampled. So we have determined or measured this total pool of carbon stock values for each one of these sites. And each one of the scenarios pulls a random arrangement of numbers from that pool per number of samples. So for instance, if you were to sample this site, the PCC, 20 times and obtain just 2 samples each in random areas of the site, your mean carbon stock value would probably range from around 26 kilograms of carbon per square meter to around 11. Here are all 3 of the figures for our sites. One commonality between these 3 figures is the point at which the decrease in the max and the min carbon stock value is the greatest. You can see that generally speaking, the change is the greatest going from about 2 number of samples to maybe 6 or 7. You can see these lines are fairly steep early on, and then beyond this point, there's still a decrease, but the decrease just isn't as great. So in addition to calculating the maximum and the minimum carbon stock values per number of samples using those 20 scenarios, we also calculated the mean margin of error. So this figure shows the mean margin of error per number of samples for the PCC using a p-value of 0.05 or a 95% confidence interval. And this figure really shows just how much of an impact you can have on the margin of error by simply increasing your number of samples. So if you were to use or sample the PCC just twice using a p-value of 0.05, you would be 95% sure that the true mean falls within about 0 kilograms of carbon per square meter and about 77 kilograms of carbon per square meter. So this is a really, really wide range, and it really doesn't give you, much information about the carbon stock value of the wetland. But if you were to increase your number of samples by just 1, the margin of error would go down by about 50, and the estimation would be a bit more accurate. So here are other commonly used p-values for this figure, going from 0.01 to 0.2. This figure can be useful maybe for an individual or an organization that doesn't have the time or resources to increase their number of samples, but maybe they're dissatisfied with their wide margin of error for a given p-value. So maybe an individual or organization can only obtain 3 number of samples for a site, and they don't like the margin of error of plus or minus 20 using a 0.05 p-value. If they relax their statistics and use a higher p-value or a lower confidence interval, they can then decrease their margin of error down to about 7. An individual or organization might also have a target margin of error, in this case 5, and be curious about how many samples it would take to obtain that target margin of error. So if you want to use a p-value of 0.05, then you're looking at getting around 15 samples for a site. If you want to stick with that margin of error, but don't want to get that many samples, then again, you could always increase your p-value, and that would only require you to get around 6 or 7 samples. So the natural wetland had the overall lowest margin of error, and the restored wetland seems to have the overall highest margin of error. So one last thing I want to say about these figures, I kind of think of these figures as showing basically an uptick in both time and resources for every increase in number of samples. So really what it comes down to when deciding how many times you need to sample a site is how much time and how much money you're willing to sacrifice for a given bettering of the margin of error or overall accuracy of the carbon stock value. So to conclude, we determined the mean carbon stocks for each one of these sites. Differences between the sites do exist. However, nothing statistically significant can be said about these differences based on the wetland type. And that's due to the fact that there's not enough replication of these sites. There was a positive relationship between edge distance and carbon stock at the PCC, but only from about 0 to 30 meters from the edge. There was no relationship between edge distance and carbon stock at the natural and restored wetlands. One possible implication of this is that boundary determinations when measuring the carbon stock of the site are especially important when sampling disturbed sites. This kind of depends on what the goal of your research or your project is. If you want to, most accurately document how much carbon a particular site has, then you probably want to sample up to the edge because that's technically in the wetland based on certain indicators, and you don't want to overestimate how much carbon a wetland could have. If you're investigating random variability of carbon stocks at a site, then you might want to give some distance to the edge and stay away from it. Because as we saw, systematic variability is usually driving the carbon close to the edge. So overall, even though costs of both increasing sample size are significant, we recommend that a minimum of 5 to 6 samples are required to make a meaningful statistical statement about the carbon stock value of a particular site. If you look at what's being recommended out there by various organizations, it seems like they are also taking this approach of not recommending a firm number for how many times you need to sample a site. These recommendations are beginning to be made based on your desired level of accuracy and site heterogeneity and overall statistical constraints that you're using for your project. So I am a film buff, and I think that credits or thank you should be at the end of the presentation, like movies. So thank you to the organizations that are here and the individuals for helping out with this project. Thank you. Yeah.
Nice talk, Patrick. I'm curious about stratified sampling. So you kind of showed your distribution of samples, and then you were showing I assume that the graphs were a little like randomly distributed, but do you think there's benefit to stratified, or can you look at your data in that way?
Yeah, I think that there can be benefit to stratified sampling. Stratified sampling is definitely dependent on the type of wetland that you're working at. So the natural wetland, for instance, it does contain pretty distinct vegetation communities throughout, which is fairly common in a lot of Delmarva bays. So I could see how you could stratify points, grouping them, based on the herbaceous hydrophytic vegetation, and then maybe the woody vegetation in the center, and then the edge. But yeah, I guess that would be just dependent on the wetland type. I know some other Delmarva bays, they are dominantly wooded, and there might not be as much of a pattern in the vegetation there. But I would imagine that would probably help out in the accuracy of your estimates.
How about in your not restored one yet, like your crop field, do you think?
I don't really know how you would stratify that. I mean, distance from the edge,
Yeah.
yeah, maybe, but.
I'm kind of questioning it out in part because NRCS, well, I think they've abandoned the project, but they were trying to look at this, like how to accurately come up with carbon stocks, and they were trying to look at different stratification schemes, but they haven't gotten very far in that, so I'm just kind of curious what your thought was on that.
Yeah, so we didn't sample, based on stratification, but I mean, I don't know, there could be an opportunity to go back and maybe look at satellite imagery and try to group up these points based on what we see on the satellite imagery. But yeah.
Thank you.
Yeah.
I think my question kind of feeds off what Stephanie's saying, because I see, like, if you're gonna put something into kind of a capitalist market, and so you have this option of sampling in a center, and you know the carbon stock is gonna be higher, especially based on like perimeter area ratio of whatever your wetland is, I'm wondering if you're gonna give recommendations about, based on the perimeter area ratio based on the size of your wetland, how many should incorporate that kind of edge distance so that they're more truthful, more holistic estimates? Have you thought about like taking on a recommendation beyond the 5 or 6 and talking about this pretty interesting finding of this increase as you move away from the edge?
Yeah, I mean, a lot of people are already sampling along a transect that, starts at the edge or close to the edge and goes to the centroid of the wetland. So, I mean, that's definitely one thing you can do to try to, fully encompass the distribution of carbon at a particular site, but.
But there's no like standardized method when you're assessing carbon stocks for like trading. I'm asking that.
So different organizations basically can like take over a project, take over a carbon credit project. Like there's various different organizations out there, and I'm not sure if there is or not, but it seems like a lot of organizations are using these formulas to recommend number of samples that are based on, again, like statistical constraints and margin of error, and that's what I've seen. But it seems like there's a fair amount of
So
organizations.
number, but not necessarily spatial distribution beyond a transect. Okay.
Right.
I understand.
But like, again, with the restored and the natural wetland, it doesn't really seem like there is any systematic variability, going from the edge to the center. So yeah, that kind of a setup, wouldn't really be necessary, I guess, for all wetlands.
Okay.
Yeah.
It looked like some of your sampling sites were actually in places where there was standing water. Just curious how your methodology had to change, your sampling technique had to change when you had to deal with that kind of condition versus the dry land.
Yeah.
The drier land.
Right. It didn't change much. So we were actually fortunate enough that we started off sampling at the PCC in the spring when water tables were the highest. Then we moved on to the natural wetland, and there was a fair amount of ponded water there, maybe at most like 20 centimeters. But luckily, there wasn't a lot of open water, so we didn't have to worry about like keeping things dry. We could kind of just like rest our equipment on top of the sedges. At the restored wetland, there was quite a bit of open water, but by the time we got to that site in August, the water tables were down, and we actually didn't have to worry about sampling in those conditions much.
Is like, did you ever have to send cores down under standing water, and then how might that have influenced your sample at that particular,
Yeah,
that particular core?
there were a few instances where the core was underwater and it didn't impact it much. Like the soil just, would stay in there, and the core remover was really beneficial for that because you can only get your arm down the hole so long before you submerge yourself. So yeah, it didn't really change much. We did end up like preliminarily buying a little sled that we planned on taking out to the restored wetland in the ponded water to kind of carry our gear, but we actually didn't have to use it because water tables were down. Yeah.
So I'm kind of a variation of stratified sampling and stratified variability. Most of your carbon is in the upper 25, 30 centimeters, and it drops off quite a bit.
Yeah,
Where's
yeah,
most
generally.
of the variability? Is it also more variable up there compared to the second half, 50 to 100?
Right. I would imagine that, so we haven't looked into that, but that would be interesting to look into.
that. I think, and this is, in your situation, it might work out to take more samples that are shallow and get a better estimate of where most of the carbon is, and then take a few samples that are deep, because there's not a whole lot of carbon down there anyway. It may be less variable or maybe more variable. I'm not sure. Sometimes deeper ones are more variable.
Yeah, that would be
But
interesting.
it's a whole lot easier to take a shallow sample
Yeah.
than a deep sample. So you could maybe take 10 shallow samples and 3 deep ones or something like that and get better information than taking, 5 deep ones.
Mm-hmm. I would imagine
And
that.
I think you probably have the data that you could run scenarios like that, because you took that, you could look at variability. And if it turns out, your data showed, at least from that one example, it's probably sort of typical that most of the carbon's near the surface.
Yeah,
And so you could see, well, how much would you be losing if your error was greater or deeper?
right.
Another way of manipulating the data you already have.
Yeah.
Might give you more practical.
Right. Yeah, that'd be interesting. Thank you. Yeah.
Yeah, so thinking about your PCC site, I know you said you took 30 samples on one side of the drainage ditch, and then you took an additional 10 on the other side. I was wondering if you saw any differences in the carbon stocks on the samples from the one side or the other, and then additionally with like increasing proximity to the drainage ditch?
Yeah, so there wasn't much of a difference between, or there wasn't much of an effect of proximity to the drainage ditch per se, but there definitely was more carbon in the points that were to the left of the drainage ditch, as these points are kind of the closest to the center of the wetland. And the center of the wetland's gonna have the most carbon. Yeah,
Hey, Brian, let's take one more and then chat afterwards.
Yeah, I'll throw one comment out related to the methodology that I work on. We require randomized sampling, which means if you go out in a wetland and you land in an open area or something, in an area that you can't really sample, is very problematic. But on the other hand, you can allow non-random sampling, right? So it's a lot of these areas that you can't sample, we just tend to ignore it. Imagine there's some areas out there that you just couldn't sample.
yeah, yeah. There were a few instances where we would hit just like a stopping point, and that would probably be like some like decomposing tree or something like that, and we would have to pick up the core and move it a little bit. But I mean, overall, actually, like we didn't have much problem getting that core in the ground. The core was really an effective way to sample.
But my question is, do you know if the variability was more driven by variability in bulk density or percent carbon?
I'm not sure. Yeah, I mean, yeah. I mean, percent.
Committee, right?
I'll ask that again later.
And I'll try to have an answer later.
Do you really need to measure bulk density, could you take the upper 50s, the entire core sample and get that mass, you know the depth. So whether it's compressed or not, you know how deep the bottom is, right?
Uh-huh,
So you know the total volume of it.
right.
And if you thoroughly homogenize it and analyze it, you could probably reduce the number of analyses. You wouldn't get a profile variation, but you would get the total stop.
Yeah, you would, everything would still be there, but you would lose the detail of the horizons, I suppose.
It's really the advantage of using the format because compression doesn't matter.
Yeah.
I see them piling up outside the doors. Jared sent out an evaluation, the typical student evaluation, it's linked to the email, the announcement, so you can take some time to fill that out for John.
It'd be
If you
interesting
join
if you
us,
could
we'll be
like
over at the
3D
hall
plot
in
or
College
something
Park.
to actually-
tried.
Oh, it won't create,
Okay.
no. Because Holly did that when she had the laser.
It's not enough.